Dear participants, my name is Gab McDonough, and I'm the Educational Action Supporting Officer at ERN-EuroBloodNet, the European Reference Network for Rare Hematological Diseases, and it is my great pleasure to welcome you to ERN-EuroBloodNet Thursdays Webinars. The aim of these educational activities is promoting the interest on very innovative topics in order to stress among health professionals the cutting-edge advances in the field of rare hematological diseases. And webinars are mainly addressed on very rare diseases, complex disorders, highly specialized procedures, and implementation of guidelines. At the same time, webinar programs that are specifically addressed to patients intend to increase awareness on a specific rare hematological disease among patient advocates and patient associations. Before we start, let me share some home rules. Please note that the session is recorded and will be uploaded in the EBN e-learning environment. If you do not wish to appear, please turn your camera off and change your name on the platform. Also, please be kindly informed that your microphone is muted. If you have any question about the presentation, please write them in the chat and they will be addressed during the Q&A session at the end of the webinar. With that, today's webinar is on the clinical usefulness of next-generation sequencing by Professor Noémi Roy. Professor Noémi Roy is a consultant hematologist at Oxford University's Hospital NHS Foundation Trust and an honorary senior clinical lecturer at the University of Oxford. She is also a clinical lead at the James Lind Alliance Priority Setting Partnership on Rare Inherited Anemias. Her main interests are hemoglobinopathies and rare inherited anemias, and in particular the genetic investigation of inherited anemias, including whole genome sequencing. And she is dedicated to using patients' views to guide how research is conducted in hematology and coordinating best practice care and red cell disorders. And with that introduction, I hand the floor to you, Professor. Thank you for your time and your expertise.
Thank you very much. Hopefully you can hear me and you can see my screen. Gavin, can you confirm?
I can, yeah. Thank you very much.
Great. So it's a pleasure to be speaking today about the clinical usefulness of next-generation sequencing. Obviously, you could look at that in a whole host of different disorders, but I'm going to specifically talk about its usefulness in diagnosing hereditary hemolytic anemias, because a lot of the issues that we encounter with this group of conditions will also be applicable to other conditions. So I don't have any disclosures. So for those of you who would like a refresher or don't know about these conditions, hereditary hemolytic anemias, they can be disorders of the red cell membrane or the red cell enzymes. And I've put here also ineffective erythropoiesis, such as hemoglobinopathies and congenital dyserythropoietic anemias, because in these conditions, you do have some hemolysis. They are hereditary. You do have some hemolysis, mostly inside the bone marrow, but a little bit in the peripheral blood as well. But today we're really going to focus on the membrane and the enzyme disorders. So the red cell membrane is very important in the shape of the red blood cell, and that's because it's anchored to the cytoskeleton. And the cytoskeleton is made up of two proteins, alpha and beta spectrin, which, as you can see here, interact with each other. And then through the membrane, you've got these transmembrane proteins that link up to the cytoskeleton with these other proteins, such as ankyrin. And I've circled in here in red the proteins where if you've got a mutation in one of the alleles, meaning dominant disorders, then you get the condition. And then in blue are the ones where you need a mutation in both the alleles. So it's a recessive disorder. And what happens with these conditions is that the red blood cells lose more and more of the membrane as they go through the spleen, and the macrophages in the spleen remove them. And the macrophages also remove the spherocytes themselves, or the abnormally shaped red cells themselves. And so they have a lot shorter lifespan than normal red blood cells. For the red cell enzyme disorders, we're looking at obviously mature red cells that don't have mitochondria and that really depend on the glycolytic pathway to make energy, i.e. ATP. And the commonest disorder is pyruvate kinase deficiency, and you can see this enzyme here at the bottom of this glycolytic pathway. And then you've got the hexose monophosphate shunt, which is involved in the ability of red cells to deal with oxidative stress and to reduce the oxidized proteins. And then we've got the nucleotide metabolism. Now, if we wanted to diagnose all of these conditions, we would all love it if we had a very simple diagnostic algorithm like this, right? but actually these are very complex disorders to diagnose, and we need much more complex looking diagnostic algorithms. And the question today is, how important is genetic diagnosis in this, and where does that fit into the clinical pathway? And obviously, we all know that there's been a huge evolution in how we diagnose or how we carry out our genetic analysis. We used to use Sanger sequencing. Obviously, that was initially done with radioactive nucleotides. And then we've got capillary electrophoresis, and now we've got these super fancy machines. This is only one example of them that does a huge amount of sequencing at a fraction of the effort, although not the cost. Now, if you look at how long it takes us to go from a blood sample to giving a report, that actually takes us 12 weeks. And you might think, well, why is it taking so long? And part of that is the pathway that I'm going to show you. And I want to remind everybody that even though we are in this super modern age of genetics, at the end of the day, our genetic analysis is pointless unless we start with the patient. And so that's where we're going to start for everything. We're going to start with the patient. We're going to start taking a history, examining the patient, doing our very routine day-to-day blood tests. We're going to have some specialized tests available to us. We might or might not want to do a bone marrow biopsy, and then we're going to have our genetic analysis. So I'm going to go through these each in order. So what's our history? What are we looking at? Well, in fact, you've got to seek out different aspects of the history depending on each life stage of the patient. So you might be thinking, what's happening at birth? And even if I have patients who present in their 30s, 40s, 50s, I always start with what's happened at birth. Have they had neonatal jaundice? Did they need to go under the phototherapy? Did they ever need a blood transfusion? We're interested in what's happening as children grow. So are they a child who was often tired, often getting yellow around the eyes? Were they someone who could do the PE lessons along with everyone else, or did they always have to sit out and get more tired than everybody? We're interested in what happens through puberty. Now, as puberty is very interesting, there are some conditions that get worse with puberty, some that get better with puberty, and some that present only after puberty. For women, we're interested in what happened in pregnancy. Did they need blood transfusions in pregnancy? And you'd be surprised the number of patients who present after pregnancy, and then you find out that they were transfused during pregnancy and nobody thought to do further investigations at that point in time. We're also interested in the family history, and it's not just asking about anemia, but it's asking about gallstones. Has anybody had to have their spleen removed? Is there consanguinity? Has anybody in the family needed blood transfusions? So this is taking a history as thorough as possible. Now, we're also very interested in the family history and the pedigrees, because that's going to help us to interpret the genetic data. So, for example, here we've got a little example of a family where both parents have a very mild condition called hereditary elliptocytosis. It looks quite abnormal under the microscope with these elliptocytes in the blood film, but they're very, very mild and probably don't hemolyze at all. And they're dominant, but if a child inherits both of these dominant alleles, they can have a very severe condition called hereditary pyropoikilocytosis, HPP, which you can see from the blood film here, would have very abnormally shaped red cells, and quite often they do require transfusions. Knowing about consanguinity is important, because then you might be thinking we'd be more likely to be looking at a recessive disorder. And then if a disorder looks like it's dominant, again, that will impact how we interpret the genetic information. On examination, we're looking for evidence of jaundice. We're looking for splenomegaly. It's pretty important to look at height and bony abnormalities. Sometimes, you have to remember to take the patient's socks off. Sometimes the abnormalities are only in the toes, and that's true for CDA, congenital dyserythropoietic anemia. Some of the cardiac abnormalities, you would only see it on an echocardiogram. So that's another thing that we would consider doing an echo. We might do an abdominal ultrasound to look for gallstones or splenomegaly. And then if somebody's got high iron levels, we might be thinking about doing T2-star MRI to look for evidence of tissue iron overload. In terms of blood tests, we've got our routine blood test, the full blood count, the retic count, electrolytes, liver function, LDH, blood film, and hemoglobin HPLC. And I always kind of think, well, if we had the option to do all of these blood tests, what's the most important test that we do? And the most important test for me anyways is the reticulocyte count. And that's because the reticulocytes, that they appear in the peripheral blood, but they're really telling you how active the bone marrow is. So if you've got nice erythropoiesis happening in the bone marrow and you've got anemia, then you expect the erythroid activity to increase in the bone marrow, and you can tell that by seeing a high retic count. But if you're seeing a normal or a low retic count, that's giving you a clue that the bone marrow is not responding to anemia. So it's not a peripheral problem, it's a central problem. And so for me, that's why the retics are really the most important blood test you can do. If you've got a low retic count, you might be thinking about missing hematinics or genetic block or destruction in the marrow. And if you've got a high retic count, then you're thinking about destruction or loss in the peripheral blood. So that's my take-home message about the retics. But you've also got other tests so there's no point to do a genetic analysis, for example, if you haven't ruled out autoimmune hemolytic anemia. And so that's why we would always require to see a negative DAP before we go on to do the genetic analysis. I'm not going to go through all the rest of the blood tests at the moment. So next we would look at a blood film, and that's going to be super important again, because when we're analyzing our genetic variants, we want to make sure that the variants we want to report are going to match what we see on the phenotype. And quite often that includes what's happening under the microscope. So we've got spherocytes, elliptocytes. I've shown you already parapoikilocytosis. This is stomatocytosis. And then here in this example in the middle is basophilic stippling. In this case, it's pyrimidine 5' nucleotidase deficiency. So we've covered the first three basics you would do before you think about a genetic test. And then the next question is, well, what should you do next? Should you do these super specialized tests, which quite often not a lot of labs do? You need to find out which lab does it. They don't do it every day. You need to send a particular sample under a particular condition to a particular lab by a particular day of the week. And so it's often quite complex to do. We can do bone marrow biopsies and we can do the genetic analysis. And the way I think about it is the vast majority of these cases do not need a bone marrow biopsy. And I'm trying to encourage people to delay doing bone marrow biopsies until we can work things out to see if the patient really needs it or not. And then you're left thinking, should I go straight for genetics or should I start with these specialized tests? So what are the specialized tests? You might be doing some osmotic fragility testing. You might be doing EMA dye binding. So for some of the red cell membrane disorders, you could do ectocytometry, which is shining a laser at red cells that are being spun very quickly to look at their deformability. You might be doing enzyme assays. And the way I sort of think about it is you would do the specialized tests, they're quicker and they're less expensive. So you would do those if you need an answer that can't wait for the 3 months to get your genetic analysis. Also, as I've said, the side effects, not the side effects, the disadvantages of doing this are that you need fresh blood, so it can't be a transfused patient. We need to know when the test is, where it is, and the transport is not always straightforward. On the other hand, doing straight to genetics, well, we can use it in transfused patients. It doesn't really matter how you transport the sample because it's the DNA is going to be quite stable in white cells, but it is more expensive and it can take weeks or even months to get the results. So here in the UK, we do a targeted panel called the R92 panel. It's got about 100 genes. And on this pie chart, you can see the size of the pie represents how many genes there are compared to the others. And you can see we've got our red cell membrane and enzyme deficiency genes, but we've also got our Diamond-Blackfan anemia, bone marrow failure, congenital dyserythropoietic anemia, and various other conditions. So why does it take 3 months to get the answer? Well, the first thing to say is really that we do a lot of things in those 3 months. So we take the blood sample, we extract the DNA, we've got to quality control that DNA that's been extracted and quantify it. We then do something called barcoding and library preparation. And this is what's going to allow us to sequence about 500 amplicons all at the same time for about 30 patients. So that's a lot of different DNA reactions all happening. And the barcoding is what allows you to then pool the samples together and then be able to pull apart the data once you've got it back. Then all the sequencing happens all mixed into a big soup. We then quality control the run. We then align each of the reads to the reference genome. We call the mutations to say which ones are different from the reference genome. We annotate and filter them because we don't want to be looking at any mutations or variants that are present at more than 2-3% in the general population. And then the very time-consuming bit is the variant analysis that's carried out by clinical scientists, and I'll go through that in more detail. We then discuss it at a multidisciplinary team meeting, and then we write the report. So what happens in this mysterious step of variant analysis by the clinical scientists? Well, what we do is we have to make a decision on how likely it is that the variant is pathogenic. And we use the American College of Medical Genetics classification, which is class 5 is pathogenic, and that means it's been published and there's some validation data to show us that there really is an impact of that variant on the gene, on the protein, sorry. Then we've got our class 4, which is likely pathogenic. So what do we mean by that? Sorry, class 5, that's the published ones. Class 4, what do we mean by that? We mean that even if that variant hasn't been published, we can guess that it's going to be pathogenic. So if you've got a mutation in the stop or the start codon, if you've got a splicing mutation, that means you're going to skip an exon or retain some intron, or if you have a frame shift, that's going to completely change the reading frame. We don't need to have additional data to call it pathogenic. Then we've got our variants of uncertain significance, which cause us the most problem. And it means that we try and work out whether we think it's disease causing, and we look at different aspects to give us clues. So we look at predictive software, and this predictive software is looking at two separate things. Firstly, it's looking at how much conservation there is between humans and other species. So you know that if an amino acid is the same in humans and yeasts, you can be pretty sure that that amino acid is very important to be not changed for the protein to work. But if you find an amino acid that is different in humans and chickens, then that's telling you that it doesn't really matter as much that this amino acid stays the same. So that means that if there's a mutation in that that causes a change in that amino acid, it's less likely to be disease causing. The other thing we look at is how similar or different the amino acids are. So amino acids are grouped into things like whether they're big or small or positively charged or negatively charged. So if you're exchanging an amino acid that's small and positively charged for another amino acid that's small and positively charged, then probably that's not going to have much impact on the protein. But if you're changing a small positively charged amino acid to a large negatively charged amino acid, that's probably going to disrupt your protein. So that makes it more likely that it's going to be a disease-causing mutation. Having said that, quite often we add up all of these different clues and we're still left with 50-50. We don't know if this mutation is disease causing. And so what can we do then? We've got a few options. So we can do some family studies, and I'm going to show you some examples of that. We can do some functional work, and that's some enzyme levels or EMA dye binding, active cytometry. And then there's a lot of functional work. So these are easy to do, and we can do them in our labs, but then there's lots of functional work that needs a research lab to be done. And sometimes we do cDNA analysis to investigate splice variants. So just to give you a flavor of what we do and how we try and work these out, I'm going to show you one little case. So this is a little boy that was born and very quickly became transfusion dependent. We did some genetic analysis. We used the target panel, and we found two variants in the gene PKLR. So that's the gene that codes for the pyruvate kinase enzyme. So that is, it's a recessive disorder, so we'd expect that we would find two variants. However, one of the variants was immediately a class 5. It had been published before, but the other one, despite going through all the steps I showed you, we were still left that it was a class 3, a variant of uncertain significance. So the next thing we need to do is we first need to check whether one variant is coming from each parent, that means they're in trans, or the two variants are stuck together on one allele from one parent, in which case they're in cis. Now, if this was the situation, then we would not be interested in this, because the fact that this little boy has two variants in PKLR can't explain his diagnosis. It can't be pyruvate kinase deficiency based on this, because it's a recessive disorder, so one variant has to come from each parent. So that's an easy step to do. And we were able to show that both the mom and the dad had a PK variant. But even though we showed that each parent is providing one variant, that doesn't mean that this PKLR variant is definitely disease causing, because obviously we have lots of differences in our DNA between individuals. So the fact that this has appeared doesn't prove that it's disease causing. So what we did next is pyruvate kinase enzyme assays. Now, I've told you this little boy is transfusion dependent, so we can't do the PK assays in him because we would just be assaying the PK levels of the transfused red cells. Now, what's helpful about pyruvate kinase deficiency is if you're a carrier of an abnormal PK enzyme, you would have about 50% activity compared to normal. Now, that doesn't cause disease, and the person is healthy, so they don't have a condition, they're still just a carrier, but we can detect that when we do the assays. So in this example, we were able to show that both the dad who carried the class 5 variant and the mom who carried this class 3 variant had low PK levels, low normal, 50% activity. So this now was proof that this PK variant is disease causing, right? So we're able to upgrade this this variant from class 3 to class 4 and call it likely pathogenic. Now, this is kind of a lot of work. It takes time. And one thing that I feel very strongly about is that labs should be sharing variants, because if one lab has done all this work, got the parents, showed the genetic test, showed that each gene abnormality is coming from one parent, and then done the PK assays, we should be publicizing that, because it means if another lab finds the same variant, they don't have to go through the same process. In the olden days, we used to publish these, but now these are too many. There's too many of these, and it's very hard to get publications out of these. So we need to find a good way to show this between labs. Now, if we look at our panel and how often we get a positive diagnosis, at the moment, we really only get a positive diagnosis from our panel 40 to 50% of the time. It's very variable, but it's basically 40 to 50% of the time. And when we do find a positive diagnosis, actually, it confirms at the molecular level what the clinician thought clinically, which is good. But we've still got 10% of cases where either on the clinical interpretation, the clinician said it's an unexplained anemia, they didn't know what it could be, or they did think it was a particular diagnosis, but actually it turns out to be a different diagnosis. And these 10% have really important implications, because they can have profound impact on the treatment that the patient can receive. And it's also very helpful for genetic counseling. Obviously, finding out the molecular explanation for someone's condition is still helpful for genetic counseling, especially for some of the membrane disorders, which can be either recessive or dominant. And the other thing is sometimes it's important for treatment. So for something like Diamond-Blackfan anemia, you could have some people who are siblings who have the mutation but don't show the anemia, and you would not want to use them to be sibling donors for a bone marrow transplant. So that's, again, an important reason to do the genetics. And then finally, I also think that the more information we gather about these conditions and their molecular kind of variation, we'll be able to give some genotype-phenotype correlations. And that's particularly true for something like pyruvate kinase deficiency, where already from clinical trials, we know that your genetic mutation can predict if you're going to respond to a new drug, which is a pyruvate kinase enzyme stabilizer. But this is likely, I think, to be the case for all of these conditions. For example, in DBA, whether you're likely to respond to steroids, whether you're likely to develop pancytopenia. For even things like hereditary spherocytosis, we might be able to predict in the future if someone's going to need a splenectomy, or even if they're going to respond to splenectomy. For those of you who want to hear or read more detail about this, we do have a paper that was published a couple of years ago now, a good practice paper on the use of NGS in rare inherited anemias. If you're not particularly interested in rare inherited anemias, but you are interested in the NGS side, this is still a really useful paper, because we were very careful to put a lot of practical information about how to set it up and what are the important aspects you need to think about if you want to offer this as a clinical service. And I would really encourage people to think about this, because what I've seen too much of is the use of NGS in a sort of research setting, which is very different from the use of NGS in a clinical setting. And you kind of have it's a different approach. So it's important to get that right. And then we've also got the guideline is also on the EHA app, and there's also a podcast on the British Journal of Hematology website, British Society of Hematology website. So I'm going to stop here, and then we'll take some questions. This may come as no surprise to anybody that we always start with good history and examination. We do our basic tests. We do use genomics, but we still have an important use of the specialized tests. And finally, I think it's important that we share our data so that we can gather more information more quickly. Thank you very much.
Great. Thank you very much, Professor, for the excellent presentation and for your time and expertise. As I mentioned in the introduction, now is an excellent opportunity to put your questions in the chat. For those of you that would like to ask a question directly to Professor Noémi Roy. And I'd also like to take a special moment to welcome our coordinator of ERN-EuroBloodNet, Beatrice.
Hi, Beatrice. Nice to see you.
Hi, thank you very much, Noémi, for
Pleasure.
the talk. So at least to encourage people to ask questions, I have a question.
Sure.
Of course, you were talking about something very simple, that is the number of reticulocytes. But do you use the production index of reticulocytes because sometimes you have a very profound, very severe anemia, and then you think that the number of reticulocytes is enough, but it's really not. So do you use this calculation, this sort of calculation?
The reticulocyte production index.
Yeah, that's it.
Yeah, I have to admit, I do more of a test.
Yeah, but maybe we have people with less experience in it.
I agree. So it is it's very helpful to look at that, because it's not just whether you make retics, but whether the amount of retics you make are appropriate for the depth of the anemia. So the reticulocyte production index, I think it's called RPMI. I don't know what the M stands for, but yes, I think that's a good point.
Great. Thank you very much for your question, and thank you very much for the response.
Thank you. And then the other way around, Noémi. So you have overall a picture of, let's say, hemolytic anemia, and you have few spherocytes, and it's really chronic, and you have excluded everything, and then the NGS is nothing. So do you perform then other tests, or you really go to the overall family, or?
So the first thing I would say is, actually, that's a good point about seeing only a few spherocytes. And I think sometimes people assume that the changes on the blood film have to be very extensive. And I think they don't have to be very extensive. And that's partly very much, for example, for stomatocytosis. Sometimes we don't see very many stomatocytes, even spherocytes. So if you have quite severe hemolysis and the spherocytes are just being eaten very quickly, then we don't see so many in the peripheral blood. And if the retic count is high, then, because at the end of the day, when a red blood cell comes out of the bone marrow, even in HS, it looks normal under the blood, under the microscope. It's only after it's been through the spleen a few times it's been removed. So I have to say that more and more, if I've really ruled everything out, I'm very reluctant to do bone marrows in these patients. But more and more, I've been speaking to my colleagues in immunohematology, because I think some of these are just that negative hemolytic, like acquired hemolytic anemias.
5%, yeah.
Yeah, exactly. And so my immunohematology colleagues will advise about trying immunosuppression, and then if they don't respond at all to that, then I would be more likely to go down the bone marrow route.
Thank you very much.
Great, thank you once again for your question, Professor Copens, and thank you very much for your response, Professor Roy. As I mentioned earlier, this is a wonderful opportunity to ask Professor Roy or Professor Goulis a question as well. And you can enter those questions into the chat. I'd also like to take a moment just to make a quick announcement about the next upcoming Thursday webinar. So I'll put that in the chat. The advantage of these Thursday webinars is that we have wonderful experts that are willing to just provide their time and expertise. And this is a video that will be uploaded, as I mentioned, on our YouTube channel. So if you've enjoyed this video and learned a lot, feel free to share it as well. So the next upcoming webinar is the treatment of anemia of low-risk myelodysplastic syndrome. And the speaker will be Urs Patzbecker, and that'll be on the 14th of November at 5 o'clock. It'd be wonderful if people could join for that as well. Perhaps I'll ask one more time. Are there any other additional questions that could be entered into the chat? Oh, we have a question here.
Yeah, we can see it. Any thoughts on further investigation in NGS negative cases, hyporegenerative anemia in childhood with negative genetics? So I would say, I mean, obviously, I think the story is very different in children, and I'm an adult hematologist. So I think in children, I would be more likely, if you've got hyporegenerative anemia, I would be more likely to do a bone marrow and to be thinking about Dimer-Blackfan anemia. With Dimer-Blackfan anemia, you can also do high erythrocyte adenine deaminase levels. So if they're high, that can be helpful. You can look for I mean, the advantage of doing the bone marrow would be also to look at the overall cellularity. You want to see if there's a reduction in erythropoiesis. So yes, I think in those cases we would do a bone marrow biopsy. I guess the other question is, if you feel really strongly that it's genetic and you're not finding the cause. I mean, for DBA, we know more than 25%, we never find a genetic cause anyways. But the other thing would be to do whole genome sequencing, because if that's something that's available to you, then if you can do the trio, just doing an individual makes it almost impossible to analyze. But if you've got the two parents and the trio, you can look for either de novo abnormalities or autosomal recessive.
Excellent. Thank you very much for that question, and thank you for your response. Thank you. Perhaps I will ask one last time if anyone has any additional questions for our professor. If not, I would just like to thank you once again, Professor Roy, for your time and expertise. This has been a pleasure to have you, and thank you for everything that you do.
Okay.
Beatrice, do you have any final comments? Or
No, just to thank Anaemi, of course, and all the attendants, of course, also. I hope you enjoy it, and you can read it again on YouTube.
Okay, thank you very much. Bye-bye.
Bye-bye.
Thank you all for joining, and have a wonderful evening.
Yeah,
Thank you very
you
much.
too. Thank you, Gavin.
Thank you. Good night, everyone.